So this topic I should have discussed before, but I'm going to put it under new findings because this paper just came out in Science on whatever it is, August 19, 2022. And this idea is not necessarily new, but the application is new. We'll look at other older review of older methods that have been tried as well. So this is about soybean photosynthesis and crop yield are improved by accelerating recovery from photoprotection. So slightly different than the other methods we'll look at. So the other methods focus on various photosynthetic processes, the details of which we will not get into, but in general, light harvesting by leaves is very inefficient, only about 2 to 5% or so, because they are like solar cells working at a very low efficiency, but they have large numbers, so they obviously can manage their business by having low efficiency, whereas for us, for renewable solar energy as a renewable energy source, we need to keep on increasing the efficiency of light harvesting quite a bit. Coming back to this, the abstract here reads, crop leaves in full sunlight dissipate, damaging excess absorbed light energy as heat. So they have to be in the open to do photosynthesis, but they can actually get much more light energy than they need, and it can create a heating problem which they have to dissipate. This protective dissipation continues after the leaf transitions to shade, reducing crop photosynthesis. So it has a shield that is very useful for when the light is shining and causing heat damage, but that protection stays even in shade, and the level of chlorophyll concentration, for example, may not be enough to maintain full photosynthetic level. For example, a leaf that is exposed to direct light versus the one that's in shade may have different concentrations of chlorophyll because the light harvesting in a shade will need more chlorophyll, for example. A bioengineered acceleration of this adjustment increased photosynthetic efficiency and biomass in tobacco in the field, but could that also translate to increased yield in a food crop? Okay, tobacco obviously has to be eventually phased out, I suppose, even though it's not going to happen anytime soon. Here we bioengineered the same change into soybean. In replicated field trials, photosynthetic efficiency in fluctuating light was higher, and seed yield in five independent transformation events increased by up to 33%. Despite the increased seed quantity, seed protein and oil content were unaltered. So this is not diluting the quality of food. This validates increasing photosynthetic efficiency as a much needed strategy toward sustainably increasing crop yield in support of future global food security. So oftentimes the population increase numbers are used to argue how much more food production has to be increased, and then we come back to say agriculture right now is one of the big emitters, somewhere around 26% of the total emission per year. So obviously we have to increase food yields without increasing not only emission but also environmental damage, water quality issues, and so on and so forth. So I won't show any result from that paper. Basically it shows that by this engineering they did to alter photoprotection when the light levels go down, they are able to increase the yield and maintain the food quality in terms of seed protein and oil content. Just for context, look at another paper here which I should have cited, which I should do right now. Let me copy the link into it before moving on. So this paper is a review of the various methods that have been used to alter photosynthesis or some other aspects of the photosynthetic cycle. We will see what we mean, again without going into details about the biological aspects, because that gets far away from what we want to discuss here. But you can see here, this is a summary of single targeted manipulations of Calvin-Benson cycle enzymes and their biological outcomes. You can Google this, or you can find this in this paper. It's a very complicated biological diagram, which I'm not going to get into, but we'll just read about the manipulation, which is directly into the Calvin-Benson cycle of the enzyme secretion and so on that are required during the photosynthesis. So the transgene expressed, again, you can just look at it. I'm not going to go into the details. SBPase, so that's the enzyme. Cyanobacterial SBPase, cyanobacterial FBPase, FB aldolase, transcriptolase, cyanobacterial SBP and FBPase, and so on. But if you look at the plants, they range from Arabidopsis, tobacco, tomato, wheat, rice, and so on, and soybean as well. And the functional description: tissue-specific expression 37 to 85% in increase in SBPase. SBPase. Obviously, I'm not a biologist. Okay, and what was accomplished? Biomass and yield. 42% increase in dry weight and a 53% increase in seed yield, or 30 to 35% increase in dry weight. So if you're going for biofuel and organic matter and other things, obviously these things matter a lot. For tomato, for example, up to 39% increase in dry weight in best lines. Tomato plants found to be more chilling tolerant. So they changed its properties so that refrigeration, transportation can be improved, even if it's not direct yield increase. For wheat, up to 40% increase in grain yield. For rice, higher growth rates under elevated temperatures. So global warming resilience can be increased, and so on. So I won't read all of these, but you can see for tobacco, 10 to 30% increase in dry weight at ambient CO2, with a 70 to 120% increase in high CO2. So even with increasing CO2, we want to keep smoking, then this is the way to do it. What else? Lettuce, 60% increase in fresh weight. And soybean, under ambient CO2, elevated temperature led to reductions in seed yield. Under elevated CO2 and elevated temperature, seed yield was maintained, while the wet weight under showed 11% and 22% reductions. Okay, so I won't go into details of the biologies, but I'm just giving you a look into how geoengineering of plants and photosynthesis has been tried, with results in the field looking very promising. Summary of single targeted manipulations of photorespiration, electron transport, and putative carbon transport and their biological outcome. Again, manipulation in photorespiration, electron transport involved in photosynthetic processes, and carbon transport. So the transgene expressed are mentioned here. The plants are like before, and functional descriptions are here. And again, biomass and yield changes, 50% increase in dry weight and no increase in seed yield for Arabidopsis. Arabidopsis. There you go. Okay. Tobacco, 13 to 30% increase in dry weight. Obviously, tobacco is mostly consumed after being dried and processed, of course, with all kinds of manipulations to make you addicted. But let me not go there. Another method, over 50% reduction in leaf area throughout the early growth phase. For let's see, rice, 17.9% increase in biomass and increased plant height. Soybean, in ambient CO2, a 30% increase in dry weight and a 30% increase in seed yield. up to 35% increase in dry weight and 6% increase in seed mass in elevated CO2. So over evolutionary timescales, CO2 and temperature changes have led to C3 and C4 kind of plants and so on. But now we are increasing temperatures and CO2 at a very rapid rate compared to evolutionary time scales. So we are now engineering the processes within plants that are already there, evolved, to see how they can respond to increasing temperatures and CO2. This is the summary of manipulations in Calvin-Benson cycle regulatory mechanisms. So the regulatory proteins are manipulated and the genes targeted are here, the plants are here, manipulation details are here, and phenotypes. So now instead of looking at biomass and yield, you look at how the phenotypes changed or the expression of the genes based on environmental conditions or these Calvin-Benson cycle regulatory mechanism manipulations. So reduction in Rubisco amount, reduction in CO2, so in rice, reduction in CO2 assimilation, increased rate of photosynthetic induction by light, and if you go back to tobacco here, reductions in PRK and GAPDH activity, reduced photosynthetic CO2 assimilation, and so on. So you get the idea that plant functional type level manipulations are being done to look for increased resilience to warming temperatures and increasing CO2 levels, but also other processes like protection against photoprotection, so reducing photoprotection impacts under low lights to increase crop yields. Are these all safe? Well, that's a separate question and we won't get into that. And when you talk about genetically modified crops or organisms, there is a lot of debate about what is safe and what is not safe, but it's clear that since the beginning of agriculture, we have always genetically modified the crops we are growing anyways, due to domestication or grafting or selecting for certain properties of the yields, plant properties, and so on and so forth. Nonetheless, it's interesting where this is all headed and why this is part of the solution that is needed for meeting food demand into the future. Is there going to be a tobacco demand increasing in the future? Let's hope not. Okay.